This method can help answer several key questions involved in the production of human milk oligosaccharides. First, how much of the product do I have? And second, what is the specific carbohydrate linkage? The goal here is to simplify and speed up the process of quantifying and characterizing these human milk oligosaccharides. This method has two advantages over the current state of the art. First, it's amenable to high-throughput screening. So you can imagine testing thousands of different conditions or perhaps even a library of synthesis enzymes in one day using this technique. Also, it uses the unique substrate specificity that nature has evolved in enzymes to give us information about the carbohydrate linkage of those oligosaccharides.
We believe that this method can improve the production of human milk oligosaccharides, which in turn can lead to better quality infant formula that more closely mimics human breast milk and help close the gap between breastfed and formula-fed babies. User discretion is recommended when deciding what method to use for lactose removal, based on cost, throughput, and equipment availability. Care should be taken to ensure optimal lactose removal.
The day before the experiment, seed 5 milliliters of LB medium, supplemented with kanamycin and carbonicillin, from a fresh bacterial colony using sterile techniques. Incubate all cultures overnight at 37 degrees Celsius with agitation. The next day, transfer 50 microliters of an overnight culture into 5 milliliters of fresh LB-M9 media with kanamycin and carbonicillin. Incubate at 37 degrees Celsius with continuous shaking until the culture has reached an OD600 between 0.5 and 0.7. First, transfer the overnight culture